Okay, so onto the esophagus. So let's take a quick look at the microscopic anatomy. We'll see that we have these different layers. We're going to see these repeated over and over in our digestive tract. We have the mucosa, the submucosa, the muscularis, in this case made up of 2 layers, and then the adventitia. The mucosa is made up of non-keratinized stratified squamous epithelium, so that's what you're seeing here. Lamina propria, which is just connective tissue underneath. And the bottommost layer of the mucosa is always this muscularis mucosae. And that's sort of this darker pink band right on this image. On our slides, it will be a much thinner pink band, but it will be, I think, a fairly good sharp contrast between the mucosa and the submucosa underneath it. So that's how you determine where you are. Anything above this muscularis mucosae is mucosa, anything below, submucosa. In this image, and in the esophagus, we have a circular layer that's going to circle around the tube, and then a longitudinal layer that goes longitudinally down the tube of the esophagus. We'll see we have one additional layer in the stomach, and then here is the adventitia, which is connective tissue that helps connect it to other organs and nerves, blood vessels in the surrounding area. Now, when you swallow, as your larynx is lifted, the bolus of food, so that little ball of food that you're swallowing, enters the esophagus, and then these circular muscles are going to contract in kind of a rhythmic fashion. Longitudinal muscles contract to kind of shorten the tube, and that's how basically peristalsis works, and the bolus makes its way into the stomach. This lower esophageal sphincter will need to open to allow the food in, and we'll see that for most part that happens. We'll see later on toward the very end of this series of PowerPoints things that can go wrong and what that can feel like. It doesn't take very long for food to travel, couple seconds for both food and liquids. Okay, so the stomach then, it mechanically breaks up food. It liquefies foods and begins chemical digestion of protein and fat. Like, remember that lingual lipase is going to start its actions here. Now we don't call that a bolus. What leaves the stomach is not bolus, it's called chyme. So just chyme. The stomach is not a site of major nutrient absorption. That role is the small intestines. So again, its main job is to do breaking up of food, the liquefying of food, and starting the chemical digestion, not absorption of nutrients. A few things that will get across the stomach, aspirin, some drugs can get across. And then, as I mentioned to you, you'll see that there's an extra layer here of smooth muscle. So we have the longitudinal layer, we have a circular layer, and then we have an oblique layer. So just one extra layer here. Let's just take a moment for some of the other main areas of the stomach that you'll see in lab 2. The cardia is this region right about in here. The fundus is this sort of domed top part here. The body is basically all of this sort of in the middle here. And then this curved portion of the stomach is called the pylorus. There's a sphincter at this end as well as up here. The stomach has folds in it. These are called rugae. Those folds will expand to allow for more food to be stored in the stomach. You'll notice there's a curve here, and there's a curve here. This is called the lesser curvature of the stomach, and this is the greater curvature of the stomach. When the stomach is empty, it's basically about the size of a large sausage. So again, as I mentioned to you guys, those gastric folds or rugae are there to allow for stomach to fill up more with food. We talked about these parts of the stomach. And as food leaves, it doesn't all leave all at once. So it'll leave in like these small little spurts into the duodenum. So let's just talk about that for a quick moment. Right, so food is coming here from our mouth, it's coming down our esophagus, so far pH 7, coming into this area pH 2. Now, the stomach is going to have some special lining to it. We're going to see that in about a slide or so. That is going to allow it to be able to tolerate that very low pH environment. That is not going to be the case for your intestines. And it can't be that way for your intestines because in order to have nutrient absorption occurring, you have to have a very thin sort of boundary between the food, or the chyme, I guess, and the blood vessels, right? So you can have that transport and the absorption occurring. So whereas the stomach will have a special lining to prevent its digestion, the intestines can't. So long story short, the acidic chyme that is coming from the stomach quickly has to be neutralized in the duodenum. So that means only a little bit leaves at a time, so that pH can be neutralized and absorption can begin. Okay, so hopefully that made sense to you. As I mentioned earlier, there's parasympathetic and sympathetic fibers that are going to innervate your stomach. Again, rest and digest, parasympathetic, fight or flight, sympathetic. Basically what this says, all the blood from the stomach is going to enter something called the hepatic portal circulation. This is important. Your liver tries to filter as much of the blood as possible coming from your stomach and your intestines before that blood reaches the general circulation. That way it can detoxify anything that might be harmful to other organs or the brain. A classic example of that would be alcohol, for example. The alcohol that you drink, some of that is detoxified in the stomach and then in the liver before it ever hits circulation. Now, obviously, some of it does because people get drunk, but it's far less than if it was injected either directly into the bloodstream or if it was absorbed, for example, through the rectum, which unfortunately some people do. Anyway, so the histology of the stomach, again, we have our same layers. We have mucosa, submucosa, muscularis, serosa. So in the mucosa, we have these gastric pits. So that's just these kind of like holes. The actual epithelial tissue lining the gastric pits is going to be simple columnar epithelium. All of this kind of in between is lamina propria. Down here, where we're actually going to get into the secreting portion of the gastric pit, is going to be the gastric glands. And then here you'll see that thinner muscularis mucosae. You'll see like in these gastric pits, we'll see blood vessels, we'll see lymphatic tissue as well. In the submucosa, it's mostly connective tissue. And then again, we see those different muscle layers, oblique, circular, longitudinal. Oh, excuse me. Okay, taking a closer look at the gastric glands, what we'll notice is that at the surface we have mucus cells, and these cells are very important. So these mucus cells produce mucus, as you would expect, but they form a fairly thick layer of mucus in the stomach, and that is what helps prevent it from being digested by the stomach acids. Two of the main cells that are producing digestive enzymes are the parietal cells and then the chief cells. The parietal cells secrete hydrochloric acid, as well as something called intrinsic factor. The chief cells secrete pepsinogen, chymosin, and lipase. We'll talk a little bit more about these in a slide or two. There are also some dividing cells that help replace any cells that are lost. And then there are something called G cells. We'll talk a little bit more about these in a few slides. So these are secreting hormones and paracrine messengers. So these are messengers that are affecting cells nearby. They're also going to secrete something called gastrin, and this gastrin is going to help stimulate parietal and chief cells, as well as increase the movement or the motility of the stomach. Okay, so here we're taking a look let me kind of try to orient you in this diagram. So this cell right here is a parietal cell. It's just a super up close view of it. This is a lumen of the stomach, and then here's a blood capillary on the other, like on the outside of the stomach. Or we can kind of think of that like what we're looking at is we're down in here, and that blood vessel is right here. Okay, so going back to this image, these parietal cells are going to be producing a lot of this gastric juice a day, so 2 to 3 liters. I mean, think about that. We've all got like liter bottles around. That's a good amount of gastric enzymes. These parietal cells are going to have something called carbonic anhydrase. And basically what this carbonic anhydrase is going to do is it's going to move this chemical reaction to the right, so going from carbon dioxide and water into this direction very, very, very quickly. So for the most part, so this is basically how you're producing hydrochloric acid in the stuff by parietal cells. Well, we have hydrogen and we need chloride, right? Those are 2 things that make up hydrochloric acid. So to get the hydro part of it, you just take the carbon dioxide and water, you quickly convert that into bicarbonate and hydrogen ions, right? And that's going to be through the help of this carbonic anhydrase. That's what you're seeing in this equation here. And then what the parietal cells do is they take these hydrogen ions and they're going to actively take them and they're going to pump them into the lumen of the stomach. Okay? So they don't put together the hydrogen and the chloride in the cell itself, but they take the hydrogen by itself and they'll pump that into the lumen, and it's active transport. You can see there's an ATP. And then what they're going to do is, again, the other result of this equation is bicarbonate. So they take that bicarbonate and they go, okay, you know what? I don't need that. What I need is chloride. So I will trade you a bicarbonate for a chloride. So it's called chloride shift. Sorry, it's called a chloride shift as we change or exchange a bicarbonate for a chloride ion. And so what the cell then does is it takes that chloride and it pumps that out into the lumen. So now we have the hydrogen, the chloride, they are attracted to one another, and that's where the acid is actually formed. Now, to me, this is absolutely ingenious, because what this means is a cell can produce acid without digesting itself, right? The one other thing too that's occurring here as we're pumping out all this bicarbonate, that ends up being picked up by the blood vessels, and this bicarbonate has a fairly basic pH, right? This is basic. So there's something called an alkaline tide, where the blood pH temporarily increases. So let's say like you're right around dinner time or right around when you're producing particularly a large amount of stomach acid. If you had like a little meter stuck into your blood vessels that's measuring pH, you would notice that as you're producing particularly large amounts of stomach acid, your blood goes slightly more basic with an alkaline tide, and then it's quickly corrected, but you'll see this little kind of tide, like a little wave. Okay, hopefully that makes sense to you. If it doesn't, please let me know. Okay, so why is hydrochloric acid so important? Well, for one, it's actually going to activate pepsin. So we mentioned back here that chief cells are secreting pepsinogen. Now look, every time you see something, I think I mentioned this to you in an earlier lecture, anytime you see this ogen, that means it is like the precursor, it is not the active form. So the chief cells are secreting pepsinogen, again, inactive. This is important. So again, the cell is not digesting itself. But as soon as it hits that hydrochloric acid, it is activated. That pepsinogen is cleaved or broken into pepsin. And that pepsin is going to be the active form that will start breaking down proteins. So that's what pepsin is doing, breaking down proteins. As I mentioned to you earlier, lingual lipase will activate in the like that acidic pH of the stomach. The hydrochloric acid will also help break up connective tissue, plant cell walls. It also will convert basically ferric ions or iron ions into a form that is easy to be absorbed and used for red blood cell synthesis. And that low pH also helps destroy bacteria and pathogens. It's pretty simple. So again, protein digestion is beginning in the stomach. Again, the hydrochloric acid is going to denature or unfold protein molecules. I don't know how aware of this you are, but protein molecules are a very complex kind of ball of protein molecules, I guess. But they're like a complex, well, think of like a ball of Christmas tree lights kind of thing, and their specific shape allows them to do their thing. If they are unraveled or unfolded, they will no longer function, right? Again, hydrochloric acid transforms pepsinogen into pepsin. Fat digestion is going to, continue. It basically, it begins here using gastric lipase that is going to split triglycerides in milk fat, as well as a lingual lipase, as well as the chymosin that we mentioned earlier, secreted by the chief cells. So this lipase from the chief cells, along with the lingual lipase. Well, really it's working on milk fats is what it's best suited for. So all of those, especially this gastric lipase, I should say, as well as the chymosin, they are most effective at pHs of about 5 or 6. So they work very well in infant stomachs. So they still work in the adult, but just not quite as well. So that's one of the reasons, not the only one. There's also like a downregulation of lactase, which helps break down milk fats. But as we get older, it also contributes to like lactose intolerance. Okay, again, as I mentioned, hydrochloric acid kills microbes, and also that mucus cells are producing that thick layer of mucus that helps prevent it from being digested. Okay, and again, some things can get through, but very little absorption is actually occurring in the stomach. And one last little bit of regulation on gastric secretions. There are 3 phases, 3 general phases. The first is something called a cephalic phase. This is basically the stomach is starting to secrete digestive enzymes even before food has entered the digestive tract. Okay, this is you hungry walking into a restaurant smelling the amazing smelling food, and we've all had that where your stomach kind of even growls because it's getting ready to get food in it. So it's already activating. So just thinking about food, smelling food, will activate the release of digestive enzymes. Then there's the actual gastric phase, and as you can kind of see here, there's food in the stomach will also activate further secretion of digestive enzymes, and this is done by stretch receptors. There are 2 main hormones that will help the increase of digestive enzymes, and that is gastrin and histamine, right? These will all feed back to increase our secretions. And then there's what's called the intestinal phase. So again, a little bit of this food will start getting into the small intestine. And you'll see intestinal gastrin, which is a little bit different. It's a little bit different than the gastrin being released by the stomach. However, the function is the same. It briefly stimulates the stomach, but then you can see secretin, something called a gastric inhibitory peptide, as well as something called CCK, are going to inhibit the stomach to prevent it from releasing more food into the duodenum, into the small intestine, mostly because those digestive enzymes need to be neutralized, that pH needs to be neutralized before more gets in here. Because again, you don't want to start digesting your own small intestine. Okay. And then you can see sympathetic fibers will suppress activity. Parasympathetic will stimulate the stomach, which is inhibited, right? So again, parasympathetic will activate your stomach, and then the sympathetic will basically suppress it. All right, that is it for the stomach. If you have any questions, please let me know.